commonly used pharmaceutical dosage forms on the market due to a long list of advantages that they have to offer compared to other pharmaceutical dosage forms. These advantages are mainly related to the facility of use for the patient. So tablets and capsules are very easy to carry, to handle, and to store, as they are light, compact, and quite stable. Moreover, they often come in the right unit dose, and they do not require any dosing by the patient, which means that they can easily be self-administered. Typically, they also have a quite long shelf life and are relatively cheap to produce, which are, of course, important factors for the pharmaceutical industry who is producing these medicines. And on the other hand, also unpleasant tastes or odors of active ingredients can be masked by the coating or the encapsulation of the tablets and capsules. So due to this long list of benefits that are inherent to solid unit doses today, even with the rise of biopharmaceuticals, which are gaining a lot of importance, about 2/3 of all prescriptions are still dispensed as solid dosage forms, and more than half of them come under the form of compressed tablets. So we will focus on this area today. Even though, as I've said, economically, the cost of production of pharmaceutical tablets and capsules is relatively low, the manufacturing of these solid dosage forms is subject to strict rules and requirements, as the pharmaceutical industry is a highly regulated environment. The United States Food and Drug Administration and the European Medicines Agency, for instance, vigorously inspect and evaluate the manufacturing processes and the control strategies for all sorts of pharmaceutical dosage forms. And they have defined a laundry list of tests to evaluate the critical quality attributes of each dosage form to ensure compliance with their standards. In the case of solid dosage forms, such as tablets and capsules, most of the or some of the most important critical quality attributes can, for instance, be the blend uniformity, the individual tablet or capsule mass, the appearance, the assay, the particle size distribution. For tablets, we will also focus on thickness and hardness of the tablets. And one very important factor for both tablets and capsules will be the content uniformity, on which we will focus during today's webinar. Within this context, the regulatory agencies and organizations have defined a large number of standards, but they have also created some guidelines and some directives which aim to help the pharmaceutical industry to meet the requested quality criteria and to achieve regulatory compliance. Two of the most relevant guidelines for our domain are shown here. So the first one is the PAT initiative that was introduced by the United States FDA in 2004, with the aim of encouraging the pharma industry to integrate quality directly into the products rather than testing it afterwards, after the production. So they proposed a system for designing, analyzing, and controlling manufacturing processes through timely measurements of the critical process parameters, which in their turn will affect the critical quality attributes. So the idea is to ensure the quality of the final product by controlling each stage of the production process based on in-depth knowledge of and understanding of the behavior of the product during or throughout the whole production process. And this then avoids the need for final testing at the end product after the manufacturing process. So this is in contrast with the traditional production approaches, where the process itself was often considered as a black box, and the release to the market could face significant delays because the control strategy was based on performing sampling on a small quantity of product that was then subjected to laborious offline analyses, which were moreover often destructive. So this is a small reminder of the PAT initiative. And then on the other hand, 10 years later, the European Medicines Agency also published a very interesting guideline on the use of NIR spectroscopy that lists the requirements for new submissions and also for variations to existing processes for all applications in which NIR spectroscopy is used for either qualitative or quantitative analyses or as a process analytical technology tool. So these are two guidelines which will be very important in our domain. The focus of today's webinar will be on efficient manners to get such tools in line. Since the launch of the PAT initiative in 2004, as I've said, and the presentation of this framework, a lot of studies have been carried out, and many of them quite successfully, at an academic and an R&D level. But the broader inline application on the production scale still seems to be prone to some obstacles which are limiting the large scale use and the full potential of process analytical technology and quality by design are hence not really exploited to their full potential at the moment. In this presentation, we will focus on near-infrared spectroscopy, and more specifically, we will discuss the use of spatially resolved spectroscopy, or SRS, as a process analytical technology tool for inline quality control. So spatially resolved spectroscopy, or SRS, is a technique combining multi-point measurements to enhance the information by the use of scattering phenomena. It can offer a balance between classical single-point NIR spectroscopy, which can lack information, and hyperspectral imaging, which is a highly informative technique, but which is often too time-consuming as the resolution is too high to be able to perform fast inline spectral acquisitions at a speed of hundreds of thousands of tablets per hour. So SRS can be considered as a simplified hyperspectral imaging system where fibers are linked to a two-dimensional CCD detector to collect the NIR signal from the inspected sample at discrete image pixels. So that allows to provide high-speed inline measurements in a variety of production environments and will be easier to integrate also than a hyperspectral imaging unit. Indatech has a patented technology which is called SAMSPEC, which stands for Spatially Advanced Measurement by Spectroscopy, and which represents a multi-point SRS probe that can easily be integrated inline to contribute to the quality by design and real-time release approach of diverse unit doses. So for instance, in the case of tablets, the probe can be mounted on top of a conveyor belt for final dosage inspection, or it can also be integrated directly into the tablet press for inline process control right before the compaction of the tablets. Due to its patented design, the SRS probe offers the advantage of being able to measure different path lengths and different depths into the sample, which allows to measure larger volumes in comparison to classical NIR spectroscopy. As I've stated before, this simplified hyperspectral imaging system also allows for high-speed acquisitions, and by moving the sample underneath the probe, for instance, or in front of the probe in case of inline acquisitions, multiple acquisitions can be performed on one and the same sample, and this further increases the surface information that we can obtain, and to a certain extent also allows to take some heterogeneity or potential heterogeneity of the sample into account. Furthermore, the design also allows to do more than just a chemical prediction of, for instance, the API fraction in a tablet, but this is somewhat outside the scope of this webinar and would maybe lead us too far. For today, as I've said before, there are a number of factors limiting the wider application of such NIR PAT tools on a production scale, even though many studies have demonstrated their potential at an R&D or a university level. Among the most relevant, I think we can state a difference in objective and mindset maybe between the R&D and the production department of pharmaceutical companies. So where research and process development teams are typically eager to enhance the process understanding and are willing to play around with certain manufacturing parameters in order to study critical quality attributes or more importantly, study critical process parameters and get to know their impact on the critical quality attributes. The production team is typically more focused on the rentability of the production and the production of very big volumes and will be more anxious about modifying machine parameters or playing around with process process settings. So even though in the long run this might turn out to be beneficial, at this very instant it implies risking to lose time and money, which often they cannot or they are not willing to afford. Moreover, secondly, changing the control strategy from a traditional sampling approach using offline laboratory analysis to innovative PAT tools is certainly quite a complex and a demanding process. So this change in mindset and culture also requires time and resources, both from a hardware and a human point of view. So on one hand, there might be the price, the budget, and the availability issues to get these new technologies in place. And on the other hand, there might also be a lack of skilled personnel, which is familiar with this innovative techniques, and it may also complicate the registration process for regulatory compliance, especially in the beginning. Lastly, I think another very important factor is the fear of the unknown. If production is currently in the comfort of a stable situation, which seems under control as there have no recent difficulties been encountered, the motivation to go and explore new approaches, even if they seem very promising, will still be limited. And this is also enhanced by a lack of experience about the stability and the maintenance of such new equipment and instruments, and the fear of maybe also revealing unknown issues. So the idea of today, or the concept that we want to present to you today is reaching out to the production scale through the use of inspection machines. So the common goal of Indatech and Pharma Technology on this project was to provide a flexible tool for inline quality control of pharmaceutical dosage forms through the development of modular automated inspection machines for both tablets and capsules. The idea is to offer a modular yet industrial approach that can easily be used in both the R&D and the production environment. So this can facilitate the transfer of technology from the R&D department, which was willing and eager to explore the potential of the technology, to the production environment where time and resources are limited. The full automation and the easy integrations should also significantly reduce the risk of losing time and money, while the required resources should be reduced by the easiness of the transfer, the compatibility with different existing production machines, and the simple and intuitive user interface. So I will now leave the floor to Freddie, who will present to you two modular automated inspection machines, the CUV20 and the CU120.
Thank you very much, Davinia, for that very nice introduction. As Davinia said, today I will present two machines that host NIR SRS technology for API fraction prediction. And these machines are designed to inspect every single tablet on their critical quality attributes. We have the CUB20, that is a mobile automated NIR PAT tool. And we have the CU120, that is the high-speed 100% content uniformity inspection machine. So let's go through the typical applications of the CUB20. The CUB20 inspects every tablet on their API fractions for one or multiple API formulations. The unit is mobile and can easily pass through conventional doors, and the CUB20 can also be used offline, at line, or online. So for the typical offline use, we have process development tasks, process stability studies, clinical batch testing, or global batch quality proof. The CUB20 can also be used at line when you need to validate new manufacturing processes or when you need real-time decision. And because it's mobile, it can serve also multiple lines. The CUB20 can also be used online for automated sampling, standalone or combined with other pharma technology equipment like a deduster or a 4-parameter tester. Let's now have a look to the typical application of the CU120 machine. So the CU120 inspects every tablet on their content uniformity for one or multiple APIs formulation. The machine performs single reject of every out-of-specification tablet. The CU120 can be used inline or offline, and the embedded 4-parameter tester allows also to perform a classic samplings alongside with the 100% inspection. Inline inspection allows you to check and sort 100% of your production batches, either conventional or continuous manufacturing. It shortens drastically your rejection strategies, and the embedded tester allows the CU120 to add a conventional sampling and physical testing report to the 100% inspection report. So the CU120 can also be used offline for global quality proof, for counterfeit sorting, and for batch recovery. Offline as well, the CU120 performs conventional sampling and add a physical testing report to the 100% inspection report. So let's discover the Cube 20 and his 3 main functional blocks. So the first block performs the dosing of the product one by one. The products are dropped in the hopper. A vibrative feeder takes care for dosing and aligning the products, and they go out on a single row to end on the conveyor. The second block takes care for the API fraction measurements. For that, the machine is equipped with a vacuum belt conveyor, a SAM spec probe and spectrometer, and a wide reference tile and position mechanism is incorporated as well. The last block takes care for the sorting, and for that the machine contains a rejection valve, a reject confirmation curtain, and a collection box. Now let's have a look to the CU120 and its 5 functional blocks. The first block performs the dosing and alignment of the product. So there is a hopper when the product is dropped in. There is a linear feeder equipped and combined with the centrifugal ball for high throughput dosing and alignment of the tablets. They go out on one single row to end on the conveyor. The second blocks take care for the API fraction measurements. This is identically the same hardware as for the Cube 20, and the machine is thus equipped with a vacuum conveyor, a SAM spec probe and spectrometer, a wide reference tile, and a probe presentation mechanism. The third block takes care for the mass measurements, and for this we have developed a GMP fast and precise 3D microwave sensor. The next block takes care for the sorting. The machine contains 2 high-speed sorting valves and 2 reject confirmation curtains. The one sends single tablets to the reject bin. The other samples and sends single tablets to the physical reference measurement station. Here is the physical reference measurement station. It hosts a 4-parameter tester. The tester weighs automatically the samples for initial and rolling calibration of the microwave sensor. The tester performs also the measurement of other physical parameters like thickness, hardness, and diameter during production. So the CU120 hosts 2 measurement technologies. The one predicts the formulation fraction in APIs. The second one predicts the individual mass of the tablets. And as a result, we get the strength of each single tablet predicted inline. Let's now have some words about the microwave sensor itself. So technology is not new, but it's the first time that is applied for this purpose in pharmaceutical equipment. Our sensor is built around a closed resonant cavity, and there is no microwave emission to the outside. The resonant microwave fields are along 3 directions. Only 2 are shown on the slides. And also there are no risk of changes to the product as the microwave power is lower than a typical cell phone power level. So each tablet is sent individually in the resonator, and when the tablets traverse through the sensor, the microwave fields are modified. The sensor measures how these fields have changed, and for each measurement we observe in total 6 parameters. Between two tablets, the microwave fields recover their natural resonance frequency. The 3D microwave resonance measurements are indirect measurements, so it requires a calibration, and it is a model that will link the measured microwave changes to the real mass of the tablet. For this, during the recipe creation, 20 tablets are sent one by one through the sensor and diverted to the 4-parameter tester. For each measurement, we observe 6 parameters at the microwave sensor, and we get the mass from the weigh cell. The software finally builds the model as a multiple linear regression over those 20 arrays. All the processes inside the machine are managed by a software layer. Both Q20 and CU120 are running under the same software platform. This means that the NIR measurements are performed on the same way on both machines because they have the same equipment and the same controls. The recipe creation interface is the most important one, as this is a place where you tell the machine what to do. It is intuitive and easy to follow. The start page is where the product data is recorded, and it is reduced to one single page. After the product data input, the user is guided through the sequence, and when required, the machine asks for inserting one or a series of tablets, and the machine learns about the product itself. For the modeling steps, the mass prediction modeling is fully automatic. The chemometrics are done outside of the machine, and therefore we have implemented a set of tools that have been designed to encode your DOE and to collect spectral data and to export them on a USB memory device. Now, Davinia will show you how spectral data can be processed by an industrial case study.
Yes, so we will now present to you the summary, more or less, of a use case that demonstrates the potential of these technologies on industrial pharmaceutical tablets. So the inspected tablets were kindly provided by a pharmaceutical company and contain diclofenac as active pharmaceutical ingredient. They have a round shape of 10mm diameter and are bevel edged. Their nominal mass was about 350 milligrams, and 87.5 milligram of API is targeted as the 100% label claim. This use case is also described in detail in a peer-reviewed scientific publication, which can be found via the link that is displayed here if you want more information or more details. So in order to predict the API fraction of the diclofenac tablets directly inline, a partial least squares or PLS regression model was developed by regressing the NIR spectra from the SumSpec probe inside a CU120 machine against reference values obtained via HPLC on a set of calibration samples which had varying API content. The calibration model focuses on the spectral region between 1050 and 1250 nanometers and applies a first derivative and SMG pre-processing in order to highlight the spectral differences related to the variations in API fraction between the tablets and to minimize the impact of maybe unwanted scattering phenomena on the spectra. The pre-processed spectra of the calibration tablets are shown here on the right, colored in function of their API fraction. And then on the left here, you can see the observed versus predicted plot of the PLS model with the calibration tablets displayed in gray and the validation or test set tablets shown in red. A root mean square error of around 1.6 was obtained for this model. The calibration model was developed using PLS Toolbox, the software from Eigenvector Research that is running in the MATLAB environment, and then exported using their model exporter into an XML file for external exploitation. And this allows to apply the model that is constructed directly inline during the measurement of new tablets by importing the XML file that was generated into the human-machine interface of the CU120 and CUB20 machine, as shown a bit earlier by Freddy. So a screenshot of this specific screen of the interface is shown here on the bottom right. And then I think Freddie will now explain a bit more into detail the example of an extended repetition test.
Yeah, indeed. On this slide, we can see two screenshots of the machine HMI. These are the results after a repetition test where we took one tablet and passed it 100 times through the CU 120 machine. The machine displays the mass measurements and the measurements of the fraction ratio. So this is the ratio between the fraction of the formulation that is in the tablet and the fraction of the nominal formulation. So we can see that the measurements are repeated quite precisely, and the standard deviation of the fraction ratio is below half of RMSEP that we got and we have seen on this previous slide. The standard deviation of the mass measurement is below the 1 milligram. For each measured value, the mass and the 10 NIR models, the machine displayed the trend over the last 250 measurements. So the counts and the statistics about the displacement, the display measurement as well. Here we present some results of the repetition test related to content prediction of the tablets. So the 4 samples are known, and they are taken from 4 different reference batches that we use for modeling. You have the nominal values on the top right table. The fraction is not exactly known, but it is estimated based on the HPLC analysis done on other tablets from the same reference batch. The mass has been measured on our lab scale. So the dosage calculation is based on the fraction ratio and the mass of the tablet. Each tablet has been measured 10 times by the CU120, and we see the statistics on the bottom of the slide. The prediction values are close to the reference values, and the standard deviation of the fraction ratio is below half of RMSEP. The standard deviation of the masses is around 1 milligram. But what finally matters is the amount of drug that each tablet contains, and here also the results are close to the reference values with a quite tight standard deviation. Bottom line, we can say that the machine is capable of precise and accurate measurements related to the content uniformity. That brings us to the conclusion side that Davinia will take over.
Okay, indeed. In conclusion, I think we have shortly demonstrated to you the use of NIR, spatially resolved spectroscopy, as a PAT tool integrated in automated inspection machines for fast and real-time control of pharmaceutical solid dosage forms. Freddie presented 2 modular inspection units who have integrated NIR SRS technology. So the first one, the Cube 20, can be used as a non-destructive tool for enhancing process understanding and is more focused on the R&D scale, while the second one, the CU120, combines the NIR SRS technology with the 3D microwave resonance technology, hence allowing 100% inline content uniformity inspection. Both machines have the advantage of offering high flexibility, as they are compatible with different production machines and can easily be transferred from development to production scale or from one production line to another. And in the small case study that we have rapidly presented, we also demonstrated that predictions of satisfying quality can be obtained on industrial pharmaceutical tablets. So then all that's left for me to do is to thank you for your interest and for connecting and being with us today for this webinar. We will also be present at IFPAC next week and at ECHIMA in August. If you wish to discuss more in detail with Freddie or some of my colleagues, and at ECHIMA, I think there will also be the possibility to see the machines in real life. And once again, if you are interested to have some more details about the case study that we have performed, it is described in much more detail than the few minutes that we have talked about the subject today in this publication. And else, well, we are now ready to take any questions that you might have. I will maybe, I'm sorry about that, stop the sharing, because else I cannot see the chat and I won't be able to see the questions that were asked. But if it's needed, I can reshare to respond or to clarify certain subjects. Okay, I don't know if there were any questions in the chat. Freddie, can you see the chat, or you want me to?
Yeah, I see the stat. I see 3 messages so far. There is one question. Yeah, I have to take care because moving. Wait a minute.
If not, I can ask you the question. So one person is asking whether the technology can be used on all types of tableting machines. So.
Well, yes, of course. We can here take every tablet producing from every tablet machine, because there is no really embedding of the machine in a tablet press. So you can use. We are speaking about tablets, so coming from any tableting machine.
Okay. Then there was also a question about if the process also works in tablets containing 2 active ingredients. So there I can say that we can do, or that's certainly possible. So in the human-machine interface, I'm not sure if, yeah, it was clearly mentioned, but we can upload and hence apply up to 10 NIR models in parallel. So if you want to make a distinct PLS models for each of the active ingredients or for different parameters of the tablet, and it's certainly possible to tackle these yeah, in parallel during the inline control. I don't know, Freddie, if you had any additional remarks on that question or that subject.
No, you are completely right. It might just have a small impact on the global throughput of the machine, as multiple models will be computed in series, but we can indeed handle up to 10 models in the recipe of the machine.
Yes. And then there is also a question, can these machines be integrated or adapted for injectable drug products apart from tablets? I think this one is for you, Freddie.
Injectable drug products, that means, if I understand well, more vials, and that's maybe more about lyophilizates and such products, I guess.
And in that case, I think the CU120 and CUB20 machine were not really developed with that objective. But if the question is more general, can NIR SRS technology be used for such applications, then the answer is definitely yes. I think we already have done some webinars before on that subject. So if you want, you can write me a message and then I can share the specific information on that subject, because that would be outside the scope of pharma technology.
I see a question about soft gels.
Yes.
It's an interesting topic. So far, it has not yet been tackled. We already are doing some evaluation on hard gelatin capsules. Soft gels, why not? But we not yet have tried anything on that so far.
Okay, then there was also a question about the price. So for that, I think the people in contact should be, or people should contact maybe Freddie's colleague Sven, whose email address was mentioned on the last slide. I will maybe show it again at the end after the other questions, so that you note his contact and get in touch for more details. And then, thanks for take the mean of the spectrum.
There was a question about RMS ECV in your model was 1.9, while the RMS EP was 1.6. Any chance with this lower RMS EP could be related to the training and test of the set size?
It is possible. I don't really remember by heart how many tablets were present in both calibration and validation set. It is true that since the tablets were kindly provided by a pharmaceutical company and they were, it's not really production tablets, it was from another study, the data set might not be perfect or ideal, so we had to play around so for instance the validation set is not 100% independent either, because we have the same fractions or same theoretical fractions of API, so it's more to demonstrate the principle and the possibilities, and it's already quite a nice data set because it's actual tablets with varying concentration coming from different batches, etc. But it is certainly possible that it is related to that. I do think that there were more tablets in the calibration than in the test set.
Mm-hmm.
And then there was also a question, do you take the mean of the spectra for each tablet sample when building the PLS model? So there the answer is yes. As I've said in the beginning of the presentation, the SRS signal can also be exploited in different ways, but then mostly it's because the objective is more to have some physical information, for instance, about the sample. Since here we try to predict the fraction of API, and we just want to collect a maximum of information or a maximum of sample, inspect maximum of sample. We took the information from all the fibers and averaged that out to get to one spectrum per tablet for building the PLS model, and also for applying it later on then.
Okay, I think so far we have tackled all the questions that were in the chat.
Okay, I will maybe reshare if I can do that quickly. Just the final slide, so that if anyone is interested to contact Sven, Freddie's colleague, that they can note the information, or even if you want to have a closer look at the paper, that you have the time to note the reference. And of course, if there are any other questions coming up later on, you can also always directly contact Freddie or myself, or any other contact that you might have, so that we can clarify things in more detail. And if there are no additional questions, then I think we can buckle up here.
Yeah, we thank you again anyway for listening and for being present. I have nothing more to add. Davinia told already everything about how to contact us. So.
And Freddie will be at IFPAC also with a presentation, I heard. So
yeah.
if you to have some more explanations, more in detail, then next week you can see him live if you're attending the conference. Okay, well then, thank you very much everyone for connecting, and hope to see you soon or be in contact soon with interesting applications or for other projects or questions. Have a nice day.
Bye-bye.
Bye.